All right, everybody, thanks for coming. So today our speaker is Dr. Jim Thorson. He's a statistical ecologist at the Alaska Fisheries Science Center, and he'll be talking today about causal analysis, dynamic and phylogenetic structural equation models. So let's welcome Jim.
Thank you. Yeah, thank you, Tanya, for hosting me, and Steve as well. And I'm flying out this afternoon, but it's always fun to be here. Before I forget, I do have a textbook coming out, Spatial Temporal Models for Ecologists, that goes into more details on some of this stuff. And then I'm teaching a grad class at UW in the spring quarter. And so people that are interested in following up, I guess, maybe email me in February or something to, if you want to just sit in remotely on that. Yeah, and I came here years ago. It's been a while. I don't know, it's just, thanks for having me, and it's nice to see you guys. So yeah, today I'm talking about sort of I'm kind of maybe halfway through a cycle of work on merging structural equation modeling into phylogenetic, time series, and spatial modeling. And I guess my point here is to try to convince other people that this is sort of a profitable path to go down, and see if anybody wants to collaborate. So yeah, I guess my claim is that, as a statistical ecologist and as ecologists in general, I'd say there's sort of three different sort of bins of inference that we want to make. First of all, the natural history, descriptive exercises. And this is sort of a paper I like from Ovaskainen and colleagues describing HMSC. So basically, if you've got, a bunch of different sites where you've monitored a vector, a set of species, densities, that's a matrix of, site by species counts. You can also have site by environmental condition, and then a species by trait matrix, and those are the three matrices. And there's kind of the famous fourth corner problem of how to infer, trait by environment kind of determinants from those three measurable corners. And people do that with joint models. And it's essentially a descriptive problem. What's out there? How do you map distributions? How do you attribute? How do you explain those based on phylogeny and traits? There's sort of predictive ecology, and in this case, we've got another paper that does phylogenetic factor analysis, which I'm doing as well, where here we have all of the mammal dated phylogeny and then different traits, species traits, and they try to look at dominant modes of evolutionary trade-offs for mammals, and you can use that to do predictive phylogenetic trait imputation to infer something like, missing size of a species, unknown lifespan, for instance, of a mammal given its size and its breeding, reproductive characteristics or whatever. And then and then there's also sort of the explanatory inference that we want to make. And here I switch to this sort of graphical modeling that's, path analysis. And so that, explanatory modeling is all about actually understanding what causes what. And, it's somewhat separate from, not everybody does explanatory modeling, but the point of this talk is basically to say that all of our science is intrinsically explanatory. And if you're not doing explanatory work, you're probably not doing science. You're probably doing engineering or something important and related. And it's about sort of trying to figure out what actually drives systems and what is a direct cause and what's an indirect cause. And, a lot of our kind of basic science, over the last 400 years is figuring out like what mechanisms we think actually occur and what are, how would sunspots or, the position of stars when you're born, if that doesn't have a mechanism to affect your income, we probably think that's astrology and not actually science. They're trying to sort out like what mechanisms can actually be intermediate causes in a causal chain is intrinsic to our science activity. But, basically I want to say that all of the work we're doing, these are three different kind of bins of work we do, but really all of them could be aided by bringing in explanatory analytic techniques. And sometimes that's called causal analysis, but So, and the bedrock of this, people, Sewell Wright started doing, path analysis 100 years ago. He's also the person who started talking about fitness landscapes, and we owe him a lot. As I understand it, he got into some fights with Ronald Fisher, and Fisher kind of pushed path analysis out of mainstream statistics, where it sort of has resided as sort of, so a lot of people in like a likelihood-based stats training don't study path analysis. Ronald was also a eugenicist and had a bunch of terrible opinions, so I don't necessarily think that his opinion about path analysis was any better than his opinion about other stuff. And, basically, like, this sort of strange history means that path analysis, despite being really important, is not something that everybody comes out of grad school kind of with a tool set for. So at its core, structural equation modeling is this top equation where you, instead of thinking that you've got some predictor variable and some response variable, you just admit that you've got a bunch of variables that you accumulate in a vector x, and you say that x arises from itself some set of path relationships in that my notation varies, and I always forget that hangman symbol, but the hangman path symbol, and then some exogenous variance epsilon. And if you just rearrange the linear algebra, you subtract the hangman times x from the left side and take a matrix inverse, you can see that the variance of x is this expression down here. Structural equation modeling always did this trick where they would say that, if you've got a bunch of normally distributed responses, then the sample variance follows a Wishart distribution, and nobody knows what a Wishart is. Probably nobody's ever, I never used a Wishart. Please don't use a Wishart. But you can fit that as a likelihood function, and you've gotten really far away from kind of our standard way of fitting models. And so this is the other reason people don't fit path models. Probably nobody in the room has fit a path model from scratch. But you can instead write it out as just a likelihood, like a hierarchical model. You get rid of the Wishart, you just give a distribution for your data, and you calculate the covariance. And that's something that we all could code up, in a couple hours, I'm sure. So the other reason that SEM is helpful is the precision matrix, the inverse of the covariance, which is kind of conventionally called Q, is often sparse. So if you've got the exogenous covariance is independent, you don't have to do any matrix inversion, and the path coefficients are often sparse. And so if you get these really large dimensional models, which we'll get into, you can do all of the computation really efficiently. So that's sort of a math stats reason that this is convenient. So the first kind of, I kind of worked out the math of this with Tim Messington, and not that it's complicated math, but it, it takes a while to kind of useful. And then I was looking for an interesting example, and so we collaborated with these people, part of Fish Globe. So Aurore Moreau is a brilliant postdoc in Malin's group. Romain and Bastien are other Fish Globe leads, and Malin, of course, here. Thanks for being here, and others. We sort of merged it continues Fish Life, this sort of trait imputation exercise I've been doing with colleagues over the last 10 years. And we wanted to get at, explore how this could be used to impute fish traits. So the idea is that if you've got this little schematic fish here, you might have 4 different traits. Some are environmental traits, some are more morphological, some are functional traits, and you might be able to measure a covariance among those traits. And if you are poly or Honig, or people from people who know regression modeling, you'll just start kind of daisy chaining one thing against another, and then another thing against that, and you get this whole daisy chain of correlations that you hope is sort of how you relate one thing to another. This sort of replaces it by doing it all jointly. So you define a path diagram in the bottom left where you say that species 1 temperature might exogenously drive species 2, trait 2, then those trait 2 drives 3, but 2 and 3 both drive 4. And you can work out the total direct and indirect effects of temperature on these other ones. And that gives you this hangman symbol that you can solve for the coefficients of. And while you're doing that, you might have missing trait values. So you might have a dated chronogram or like a phylogeny with known branch lengths from molecular records or molecular markers, neutral markers. And you might have some measurements of continuous traits and other measurements of discrete traits. And your goal is to fill in the missing discrete traits. So like if you've got something like, you spent $1 million, figuring out, do chili pepper spawn one time or multiple times, and then you want to figure out, does quillback spawn multiple times, and you've never done a directed study, you might say something about their similar habitat use and similar morphological characteristics and try to make an informed prediction of that missing trait information. So yeah, this is sort of a repeat of this. Like you can, this is all using what's called Gaussian Markov random fields, these sort of ways of parameterizing a big multivariate normal distribution. And you can make this Q an inverse covariance or precision matrix that arises from the modeling, the sort of evolutionary trade-offs. And then you can separately make this covariance in, and this is sort of phylogenetic comparative methods like Felsenstein, and people have been doing this for decades, but not really in fisheries. You make this covariance based on evolutionary relatedness of species, and that gives you a separate precision matrix, the evolutionary precision matrix. And then a separable covariance is just the Kronecker product of these two. So, and then you can say that joint precision defines the covariance for all species and all traits. So this is sort of a trick from spatial modeling. So we do this in fish life. We publish this in methods. If we have 53 variables and 34,000 taxa at the time, some are continuous, some are discrete. We boil it down to 26 variables. We plug it into this software, and we get, with a priori specified tree or graph. This is sort of the set of path coefficients that we estimate. So we made this graph based on sort of a little bit of like a metabolic theory, like temperature being exogenous and driving other stuff. And then, length, asymptotic length being the kind of master trait, sort of a Ken Anderson inspired kind of evolutionary model. And there's, plenty of stuff in here. SEM expresses all these covariances as regression coefficients. And so some of these regression coefficients line up with the old 90s style linear modeling that, Pauly and others did. But some of them are interesting and surprising. So like, for instance, Ken Anderson has this theory, and this is the story I always tell, that fish want to maintain a fixed adult size to offspring size. There's an optimal value for that. But that as fish get smaller, they can't shrink their offspring past a certain point. Like basically eggs, when they hatch and post flexion, they have to start being visual feeders, and you can't shrink a retina smaller than about 1 millimeter due to cell stuff. And so, the smallest size of an egg is about, 1 millimeter, where, an egg is all retina, at a certain point, which I learned in Japan at eating eggs. But yeah, so, as you shrink fish, as you get smaller maximum size, you start getting fish that have these alternative guarding or reproductive characteristics. Either you get mouth brooding cichlids, or you get guarding, stream sunfish, or whatever it is. So this sort of, length has a negative relationship with spawning type garders, where, as they get smaller, they're more likely to be guarding type. So that means that if you can measure, if you go out and find a new fish, and that it's a garter, that gives you conditional information about its maximum size. These are the kind of fun stories you can start seeing. Just to kind of play this out, we also use this archetype analysis, which Laurent Pécuchet has also used. And it basically models we have these imputed traits for all fishes. And then we run that through and make all fish a mixture of 3 different or an unknown number of traits or archetypes. And we selected 3. And you get this sort of trade-off that looks basically like the Wine-Miller-Rose trade-off, so use these imputed traits to then assign all fish kind of probabilistically to those 3 kind of periodic, opportunistic, or equilibrium style fishes. And it's and then you get the sort of trait, so the trait by archetype stuff on the left. That's how I named them, how I assigned them, the Wine-Miller Rose stuff. But I guess what I like pointing out is that I thought I knew something about like history trade-offs, because I studied like a fish there, and I studied a fish there, and I studied a fish there, but like all commercial fishing is sort of right in here. And in fact, there's all these kind of insect fish over here that have like, they have a maximum lifespan of 30 days. And, there's a whole set of fish ecology that I never think about because I do commercial fishes. And so, it's sort of useful to do these sort of exercises just to kind of recapitulate theory. So, here's the sort of comms department workup of this, and Jewett's dwarf goby, is sort of this sort of outlier on the opportunistic side that probably grows in somebody's swimming pool, or but yeah, if people are interested in sort of climate driving trade-offs among different strategies, this is sort of a complete database of that. However, fish life has gotten this cumbersome, and I think I need to refactor it, and I'd like to work with people to do that, where basically, somebody should be doing the trait compilation, other people should be doing analysis, and other people should be doing distribution, and I'm not very good at anything except analysis, and so I need to collaborate. So we kind of forked off a package called Phylosem that's on CRAN. That's sort of the simple version of fish life. So if somebody wants to run trait imputation or kind of phylogenetic comparative methods, this is sort of the R package version to do it. And this is with Wouter van der Bijl, who's a great postdoc at UBC. My kind of cartoon of this is that we often have sort of a team of people that do neutral traits, neutral markers, and then you use Mr. Bayes, and they've got a bunch of stuff. There's some geneticists in here, and so I'll try not to be too jokey about it, but I, like as somebody not a geneticist, I sometimes have no idea what they're talking about. They got all these plots, and so there's sort of a tree reconstruction that is very important that one set of people do, and we have a hard time communicating their methods because they're specialized. And then after that tree is complete, then we can use that to inform trait imputation, and that's a separate group of people, I think, that often do that. These two parts of the stack are important to work together and often don't. And so, Phylosem could be part of a larger effort to kind of, unify evolutionary biology with fisheries ecology. The package is up on CRAN, as shown by this wonderful CRAN thing that all packages have. And basically, the schematic is, you have to give it this phylogeny. In this case, this is the Fenadol database, and these are subsetted. It's from Fish Tree, which is this Rhodopsy tree that's also on CRAN. Then you've got a natural mortality rate and asymptotic links from the Fenadol database. This is like this sort of gold standard mortality database that we use in stock assessment. You could specify a relationship a priori, you put it through this model, and it outputs sort of stuff that you can automatically plot with other packages that I don't have to maintain, showing the imputed traits and an estimated relationship. So, yeah, this is sort of operational science in the sense that it's a bunch of easy-to-use packages that we could do annually if we wanted. The inputs are, APE for the tree and SEM for specifying structural equation models. And the outputs can be coerced with these different objects for kind of ease of use and kind of sharing. And the full syntax looks like this. And this is sort of the syntax I always use where you specify sort of one or two headed arrows representing an SEM. And you just pass it. This is the data set that people use in this ECM lit. And then you can plot it by coercing it to an object. So there's a bunch of different kind of evolutionary covariances that PCM uses. So there's a random walk or an Ornstein-Uhlenbeck or these different transformations, Ogle's lambda or delta. They're all different transformations on this evolutionary relatedness. And yeah, and you can do it with GLIMs. So you could have, some multinomial data, some you could have raw data for, broods and fit that as a poisson. So by kind of merging all this together, there's sort of this trait imputation package R phylopars. There's SEM that doesn't do the phylogeny. There's phylo-LM that people do for standard regression, and so on. And anyway, phylosem kind of merges these different kind of techniques for PCM. And kind of to try to claim that this is useful, again, like coming back to, stock assessment, finally, we need to know natural mortality for almost all of our reference point determination. I mean, the way it's set up currently. And it's incredibly hard to know what natural mortality is if it's anything. And so people come up with these regressions that they sort of bless and hope that work. And the goal is to have a improved way of doing that uses evolutionary trade-off information. There's also a bunch of additional work that could be done. So like ecosystem parameters, like consumption over biomass, Q over B, is sort of an ecopathic ecosystem parameter, and that has phylogenetic signal, like seabirds are like 40 and lumpfish are 1.5 or whatever it is. And so there's a bunch of ecosystem-y parameters that people haven't done the PCM trait imputation for. Same with energy density, like we do bomb calorimetry at great expense for a few species, and then we just assume every other species, we just pick the one that looks the most like it if you look at a picture and just hope it's the same energy density. And that's not a very principled way of sharing information in our process research when we do ecosystem studies. So the ways we do this, to infer missing values, sometimes we do a linear model, and that's nice because we know how to do it. We can do a nested taxonomic linear model, and this is where people want to get fancy and have a methods paper on the same thing.
I have a request online to stay on.
Okay, I'm sorry. Got it. That's why the screen was up. I forced it to be back so I could wander. But yeah, and then this, these phylogenetic comparative methods are about basically informing the covariance of this mixed model using evolutionary relatedness. So that's what this phylogenetic linear model and phylogenetic comparative methods does. And basically, I'm taking that and extending it to these sort of SEM kind of kind of causal kind of explanatory modeling contexts. So if you want to do phylogeny, I mean, it brings in information that a linear model is leaving on the chopping floor. But the people in fisheries have been doing nested taxonomic models for a long time, and it's true that that's probably good enough a lot of times. So for instance, if you look at 6 species, you look at their taxonomic relatedness and convert that to a covariance matrix, you could get a covariance matrix like the bottom there, and that would be the blockwise covariance of a mixed model. Or you could use phylogeny and the actual branch lengths to do phylogenetic, evolutionary relatedness, and it would reveal, a weak correlation between the Merlucciuses and the Lutjanids, because they're more closely related than the Ingrowlids. But there's those two covariances are pretty similar, so it's probably not going to make a huge difference. Anyway, but it gives you automatically, it gives you the ability to do very easily these sort of, which surprisingly has not been done in trait imputation. So again, Then et al., with John Honig, kind of published this mortality prediction based on longevity or growth parameters, and it's used in stock assessments worldwide. Conventionally, we do this kind of high quality one where we predict M based on longevity. And then there's this sort of fallback if you don't have longevity information to predict it from growth parameters, and that's shown there. And for instance, like Jason Cope and Owen Hamel have kind of published back and forth with Then and Honig about kind of, different variants on this. But my claim is instead, I can make this phylogenetic SEM that kind of captures both of those models. So here I'm saying that growth parameters cause or explain longevity, and then longevity explains mortality. And I don't think that's actually the evolutionary trade-off. That's not an evolutionary model, but it's a convenient way of making the model where growth parameters are conditionally independent of mortality, conditional on longevity. And that means that if I've got a longevity measurement for a species, it'll basically ignore the growth information. So it'll revert to the longevity relationship if you have longevity. If you don't, it'll use evolution to infer that missing longevity. Yeah, so and then there's a couple, again, you could exclude natural mortality and keep longevity information. That's called the kind of longevity prediction. Or you could drop mortality and longevity and you revert to that growth prediction. And I'll do this jackknife experiment to see how the two different methods. So in this case, this is a histogram of the 200 jackknife runs, dropping one stock, fitting the model, the other ones, then predicting the missing stock and comparing with the known value. Where percent variance explained should be 0 if it's good. Or no, percent variance, 100% should be good, and the error itself should be 0. On the left-hand side is the linear model, the kind of standard like 90s style of doing comparative methods. And on the right is sort of this proposed sort of easy to swap in alternative. And the top row is using longevity information, and the bottom row is dropping that longevity, so it's forced to use growth parameters. And in the top row, it's basically saying the percent variance explained is similar between the linear model and the phylogenetic SEM. So if you have longevity information, you don't need to do anything fancy. There's not any phylogenetic signal in the residuals, and you do as well with the linear model as fancier thing. But if you don't have longevity and you're using growth information, the linear model explains 39% of the variance. The PSEM explains 52. It gets about 40% more statistical efficiency. So clearly there's some evolutionarily conserved residuals that the model can condition on to improve prediction from growth parameters. And again, I can use the phylo signal package to plot the predicted slopes of this sort of random slope model. And red are rockfishes, and the right-hand side is the it's a random intercept model. It's the right-hand side is the random intercept for using longevity, growth parameters to predict longevity. Rockfishes all tend to have this positive residual in their intercept, meaning that they have a higher longevity for a given set of growth parameters. And I think anybody who's looked at a rockfish kind of prominently sees that they have kind of not all of them, but a lot of them have armoring, either wider bodies or spines on that thing on the front that I haven't taken a fish class in too long to remember what it's called. There's some spines in their fins. I remember what those are called. But yeah, so there's these sort of phylogenetically correlated residuals in their relationship between growth parameters and longevity, and that's the point of this. So using phylogenetic comparative methods allows you to condition on that to improve predictions of natural mortality. It's the sort of takeaway of that. And then we've done this in the Alaska Center. We presented this to Plan Team. I actually didn't put the Pacific Cod stuff up here, but the Pacific Cod assessment this year used this method, so sort of being used for management, and it's it's the kind of thing that. But here, if they wanted, Jason Cope is talking about doing it. That a lot of times, in my experience, people don't want to use a global meta-analysis. They want to go through and figure out what data they actually try. I mean, John, you and I were talking about this. Like a lot of global analyses are based on data that is clearly wrong if you start looking into details, including mine. I just use Fishbase. So if you're doing management, maybe you just want to build a database of data you trust and then run the same model on it. And I could get somebody set up in half an hour using CRAN to do this on data you trust. And that's probably the way to move forward with meta-analysis, I think, in fisheries is actually allow people to do sort of regionally validated versions of these models. Okay, so that's that sort of phylogenetic SEM arc. And then, having kind of convinced myself this was sort of working, we did the time series extension of that. So this is with Alex Andrews, who's in Juneau in the fall survey, sort of juvenile survey team, Tim Essington at UW, and Scott Large, who's the ecosystem branch chief at the Northeast Center. And this paper's in minor revisions at methods and has a package called DSEM that I'm trying to get up on CRAN this week. So, again, there's a vignette. PhyloSem has vignettes that replicate all the stuff I showed. This will have a vignette kind of replicating all the stuff I'll show that you could access by end of week. So yeah, some of this stuff we've talked about. I guess my claim is that every field has its own vocabulary for talking about causal analysis. Possibly with the exception of fisheries, where we don't really have a vocabulary to talk about causal analysis. We often just kind of assume we know a set of nonlinear equations and kind of, hope that it works. Sorry. So yeah, but economists have, there's tons of books by economists about causal analysis. And they often use, talk about simultaneous equations and instrumental variables and identification criteria. Ecologists have these kind of well-known things like trophic cascades that we'll talk about. Statisticians have these vector autoregressive models, and these are all different submodels that you can get from a time series extension of SEM. So yeah, kind of to put a sharper point on this, I'd say that fisheries, we often, ecosystem-based fisheries management, we often, and stock assessment, often specify these complex structural form models where we make very strong assumptions that, a cohort will survive except for what's caught by fishing and natural mortality, and that survival over time. We've got a bunch of nonlinear functions in F, but essentially it's just a Markov. I don't know why it's doing this. Yeah, but yeah, so it's a Markov model with a nonlinear F. And you can infer causality if you happen to get F right, and if you don't get F right, you're kind of hosed. And that's where that lies. Process research, by which I mean all the people paid to do, habitat studies and survey juveniles, do kind of short-term targeted experimental or observational studies, often do these kind of reduced form models where they take one thing and regress another and, hope that they get something significant. And, if you get something significant, it's often thought to claim that, X is causing Y, like, PDO might be causing recruitment of anchovies or something, And, again, it's sort of, it can work, but it often people look at across studies at a daisy chain of these. So like they'll look at a study that published that PDO affects anchovy recruitment. They look at one that PDO caused hake recruitment, and then they line up the coefficients and they say that like, oh, PDO is affecting hake more than sardine, or and if you daisy chain a bunch of these individual regressions together, you're probably not going to get the right answer at the end. And so the hope is that we can come up with a way of doing this with simple to use methods that we could do in our ecosystem status reports that are likely to get the right answer algebraically at the end about which of these is more important. Yeah, so I'm going to kind of walk through how this could kind of count these other nested submodels, and I'm calling it dynamic structural equation models. It's sort of a simple idea and surprisingly hard to find in the stats literature. So again, if we've got these simultaneous equations, we can back out the covariance and a Cholesky of it, but this doesn't have lagged effects. And so we could add autocorrelation in a time series model and make the separable time autocorrelation and SEM, and that's sort of where I started. But that only gives you one lag for the whole set of simultaneous equations, and that's not what we want either. And so instead, we can just kind of vector, we can stack the columns of this vector, of this matrix of variable by time, and define a simultaneous autoregressive process on that. And that gives us the ability to specify any set of lagged or simultaneous effects, and it's still a big GMRF. So it's essentially a way of making a non-separable variable by time interaction that allows you to specify in a kind of granular way any set of simultaneous or lagged effects in a linear time series model. And yes, that's what this package DSEM does. It it predicts missing values. It has latent variables. You can include loops and non-recursive, recursive dependencies. So if it's non-recursive, you could do a set of linear models. If it's recursive, you can't have a set of linear models to walk through the whole thing. You can estimate the effect of press or pulse experiments or initial conditions. You can do the GLIM stuff like a link or a distribution, and it's very fast because it's a big GMRF model, so it runs in a tenth of a second. It's faster than you are to read the output. So yeah, so it'll be out soon. So economists do this. So there's a guy, Klein, who is sort of the father of input-output models. Like, just on a tangent here, like the early input-output modeling. There's a guy named Leontiev who is the father of input-output models, and it got picked up by the Soviet Union actually. But they didn't really know the math that well. Like, it's hard to do a command economy if you don't actually have good algebra for figuring out how many shoes you need to produce a bushel of wheat or whatever. And so, it's maybe we'll come back to that. But Klein is famous for having sort of gotten our national accounting of economics together, kind of following up on input-output models, and I think he got a Nobel Prize for some of this stuff. Anyway, in econometric models, there's this sort of standard macroeconomic model. There's an equation for consumption and investment and private wages, and you can rearrange these equations in different ways. They're all the same expression. And they have these lagged effects, and the same price equation shows up in multiple consumption and investment. So it's not really clear that you could set, you can do this with ordinary least squares, but it's a little, there's some interesting dynamics that pop up. Yeah, and so in DSEM, it's fairly straightforward to publish this. Specify this. You just have your set of path coefficients written out as a text file, and one head of errors means something causes something, and then you specify a lag. So here, like corporate profits drives consumption simultaneously, and then there's also a separate lag. Corporate profits, maybe they save it, and then it drives private wages has a consumption effect that's the same. Private wages and government wages have the same, so you can mirror parameters. So it's sort of a nice flexible interface for defining linear dependencies, and you can fit it easily. And Klein published this time series from the Great Depression, where there's this sort of big decline in corporate savings, and they stopped investing, and that drive. And so these sort of general equilibrium models are useful to map out, if you've got a shock, like a pulse experiment where you evaporate all corporate, savings, how does that walk through this sort of ecosystem of economic variables? And so that's why economists care about these general equilibrium models. You fit it, you get the same answer using a GMRF as you do, the way economists publish, and you can get these kind of causal models down below. And obviously, I'm kind of leading up to the idea that, economists have been doing this for 60, 70 years. They also are probably more listened to in our government than fisheries, scientists. And so, maybe there's a causation there. So we could just get better models. I'm sure we'd be as trusted as the Fed. But yeah, so that's the economist story. Ecologists, by contrast, we got stuck talking about trophic cascades and, but anyway, so here we've got, we dug up this data set, Tim helped me, of sea stars affecting sea urchins, affecting kelp, and then sea otters having a direct effect on kelp. And in this case, we've got 4 variables across 12 sites. So we've got 48 variables across about 30 years of different length. So we have to map effects to be constant across space, which we can do, and deal with missing data. And what pops out is this path diagram where we've got a significant, the p-values are down below. So we've got a significant effect of Pycnopodia on log urchins and significant of log urchins on kelp. Both are negative, and so that's that trophic cascade that's significant. The sea otter piece is non-significant and positive. So you can kind of get, a trophic cascade pop out of this. Other people talk about vector autoregressive models. So, Tony Ives and Dennis and Tim Wooten and many others have been interested in these vector autoregressive models as sort of an approximation to any nonlinear Markov model. Yeah, and so, where you've got this B, which is the community matrix, and there's properties of ecosystem stability that you can get from the and it's a multivariate Gompertz model if you do everything in log scale. So here, I got data from the of wolf and moose abundance on the my kid loves Jurassic. It's not Isla Nublar, that's the fake island in Jurassic Park. It's Isla Royal, Isle Royal. Yeah, so and then if you want to specify a vector autoregressive model, you just do all of the pairwise lagged effects of the lag of 1. So that's the bivariate var specification as this arrow and lag notation, and fitting it down below. And the raw time series look like this. I mean, looking at these data, I'm not sure why people don't talk about this like big moose die-off in Isle Royal, which seems like the big story to me, but that's not what people use these data to represent. And then there's this sort of oscillation of wolves. There's a bunch of different software people have done for VARs, and they all give you the same result, confirming that DSEM can do a VAR easily, and you can plot it as a graph down below, where wolves have a negative effect on moose, and moose have a positive effect on wolves. So you get this predator-prey cycle in a VAR. I'm so sorry. Yeah, so then fishery scientists, I mean, we love to debate recruitment, and so, there's these paradigms you get of, direct climate impacts, like if you get if you spend $1 million making a numerical ocean model, the world is everything's a nail for larval drift. I don't know, but yeah, so you might think that there's a direct temperature impact on recruitment. You might think that temperature drives forage availability or predator-prey overlap. And, at my center, there's the people who have a numerical model and want to spend SEFI money, Climate Fisheries Initiative money on the ROMS model. There's the people who want to use it on the spring copepod survey. There's the people who want to use it on the summer acoustic survey. And all of these people, marshal kind of one-off process studies that suggest their linkage is the strongest. And it's, it's an important argument that we get right. Now I'll see if I can do 2 things at once. But yeah, so it'd be nice if we could make a model that allows us to sort of acknowledge that it's not just climate that is driving recruitment, but it's almost certainly something about climate driving something that fish eat or do that then affects recruitment. Can I trade off? Yeah, here we go. Yeah, so in this case, this is like an ecosystem, ecological and socioeconomic profile, like an ESP in our kind of council lingo for recruitment of Bering Sea pollock. And if we assemble all the time series for Bering Sea pollock, and do this ESP structural model, we get a model where log sea ice drives cold pool extent. It has a positive effect on cold pool extent. Cold pool extent has a positive effect on fall calanus abundance and a weaker effect on summer euphosid density. So if you have a cold year, the proportion that kind of in the ocean, there's relatively more calanus to euphosid. And then each of those has a positive effect on fall diet proportions of calanus and euphosid in a fall survey of age 0 pollock. And then those proportions in the diet then has a positive effect on stock assessment estimates of recruits per spawner with a separate Rickert or Gompert style density dependence term affecting recruitment. So there's essentially like a stock recruit model here, a state space stock recruit model. And then instead of our standard thing of using a single covariate to try to predict recruitment, where you choose whoever you like and choose their time series, you actually do this sort of structural model where you try to think through the various thing mechanisms, figure out what data you have to measure those mechanisms and measure those. So obviously this is sort of my stump speech for doing structural modeling embedded in a stock assessment. And then again you get these sort of missing values imputed. So our a lot of times our proximal causes are biological samples that don't run every year, whereas the ROMS model you can get annually. And that's, of course, the reason people want to use the ROMS model to predict recruitment. But yeah, missing data shouldn't be a problem if you actually have a proper time series model. You just sort of jointly impute and predict effects, and you'll get your kind of statistical leverage. Right? So so why care? Well, you can deal with missing values and in these sort of recruitment studies. Like oftentimes, again, like we send our calanus copepod survey data to Poland for them to do measurements, and we do a rapid zooplankton assessment count on board. And so you could have like a composite variable that's copepods, and you've got a unlagged measurement that's noisy, and then a 1-year lagged high quality measurement from Poland. And you could make sort of like a way, that you can make a way of assimilating sort of rapid and high quality biological sampling in recruitment studies. It allows these conceptual models. So like, back to Richard Levins kind of talked a lot about qualitative network models as sort of a way of getting generality. Yeah, I don't remember that paper. I forget I mentioned it. But there's a reason that he likes qualitative network models from the 70s. People still do these, like John Ream does these and Eva Plagiani, and they're sort of like a simple type of data limited ecosystem model. So we're talking about trying to go in with, like Alaska Native stakeholders and build up a conceptual model for Kuskokwim, Yukon River, salmon die-off. We can't wait for an EBFM model that takes 5 years. Who knows, that's a generation of salmon. So these kind of provide a rapid ecosystem model. And then the math is easy enough to embed directly in a stock assessment. So Colma and I are doing this for state space assessment models for Goa, Pollock, and it's sort of a generalization of like Tim Miller's stuff for dealing with missing values in covariates of state space assessment models. So here's a list of collaborators on related work, including Steve, who got me going on the PCM stuff years ago. Thank you. Thank you, everyone. Yeah. I'm sorry.
Yeah, sure. The phylogenetic approach, do you think for within species variation, local adaptation, perhaps sort of matrices of genetic distance, seems to think that could be applied in that setting as well?
Yeah, so Wouter van der Bijl, who I did this with, does a lot of within species like pedigree information, and in fact, what's called Poggle's lambda in PCM is the same as heritability if you fit the same model to a pedigree. So there's a, you know, and Felsenstein, who developed PCM, also did a lot of pedigree information. And so that, yeah, it's 100% applicable with no real change in software. I haven't done as much, you know, trees. You know, one thing that comes up is, you know, all the trees I did were species as tips. You know, you could also do stocks within species and then individual within stock, or, you know, time within stock. There's all these sort of extensions that the math is straightforward, but the software has not written itself. So it'd be
with unrooted trees.
And yeah, you can do it with unrooted. It's easy. I mean, again, it's like, you know, the math is the same as a stream network, which we're going to talk about later. And so, and that's how well, anyway. So yeah, I mean, there's another, basically, like my stump speech is that we all get trained in regression modeling, and we've been lied to. Like, regression modeling is not a good way to start thinking about statistics, because it basically leaves all of the ecology, all of the things that cause our covariates to be collinear. It makes us ignore that. So it's a training that makes us forget most of what's interesting in the way we set up a regression model. You know, SEM doesn't start by distinguishing exogenous and endogenous variables. It kind of allows you to specify that. I'm sorry, that's a rant. That kind of distracted me from what I was actually answering. But yeah.
That was really great. My question is using phylogenetic for causal inference. I was thinking, like, for example, if I have an analysis that shows like negative covariance in phylogenetics signal for two traits, and I wanted to use phylogenetic to look at maybe two different hypotheses that are two different causal pathways, and maybe compare support for these two hypotheses. I know that can be tricky depending on how models are fitted, and so I was curious if you can speak to whether phylogenetic has like functions for doing this sort of like quantitative hypothesis testing between distinct models, or if there's anything in it that helps facilitate or maybe constrain doing that.
Yeah, so yeah, so the question is about like model comparison and validation in phylogenetic structural equation modeling, I'd say, and maybe model averaging too. And Wouter, my co-author, I got to know him because he authors a package called Phylopath, and you know, which does all that model comparison stuff. So yeah, so there's a bunch of utilities built into phyloSEM that is supposed to allow you to fit a bunch of models easily, and you can use AIC to compare them. The literature on validation in SEMs is extensive, and I don't have that much to say about it. I'm not an expert on it. You know, you can do hard, assuming your model is right, assuming linearity is right, you can do, which it's not, to be fair. You can do, you can get an omnibus test, like a p-value on the conditional independence that you're assuming relative to the conditional independence of your residuals in your fitted model. And so that's the sort of SEM trick, you know, where they have an omnibus test to accept or reject your selected model. And there's sort of SEM people that are like, that's the only basis for model validation. There's other people that care about parsimony that do wald tests or AIC or whatever. That's the short answer.
So the way I understood is that the weights on your graphs are all based on like a linear relationship. So I was just wondering if you had thought about extending this to nonlinear causal variables.
Yeah, so Steve is 100% a better expert on that than me. The limited work, I'll turn it over to you in a second. The limited work. Okay, well, no, I know, but that's, sorry. Yeah, you know, the usual trick to kind of broaden the generalize a model is to embed your submodel in some set of powerful math, you know, like some larger body of math. And so you can, all the stuff I've presented, you can reframe as a solution to a differential equation. And so like for the SEM stuff, you can make it nonlinear by instead tracking advective diffusive movement across a fitness landscape. And that's what Sewell Wright, of course, must have thought about given their published interests. Now we have computers, so it's actually pretty straightforward to do low variable phylogenetic SEM for a whole tree as a movement model. And I've got software doing that. There's a few people in PCM that are interested in multimodal or other fitness landscapes, but they don't, strangely, I haven't managed to convince any of them to really care about estimating a parametric, there's a lot of theory like Mengel stuff, that suggests a functional relationship and trade-offs between traits. And so it'd be nice to be able to fit those nonlinear kind of evolution-derived models to PCM. And it's not hard to do that. It's just not what anybody does. And so I'm not going to publish a paper in a group of people who don't know me on something that they don't care about. So.
I was also wondering how do you deal with like variables that cause each other, right? Like in the moves and roll example, because from what I understand about tags, yeah, it's cyclic, right?
Well, so this can be cyclic, but you can't. Yeah, so like you could identify X causes Y causes Z, and Z causes X. That's identifiable even just from observational data. But if you want to do like an X causes Y and Y causes X from observational data, you need to fix one of them. And so that's one place where you can put in experimental data. Like if if we had a program that spent millions of dollars periodically on coral research, and we have some samples of coral, we could do models where you fit a joint model of, where corals affect rockfish density. And then you can also do experiments like putting out fake structure and seeing, experimentally measuring one of those effects. And that would allow you to infer an unobservable from an observable and an experimental effect. So there's sort of a big synthesis of observational experimental stuff that's possible with SEM. Yeah.
I have a question going back to the section. That sort of a priori information matrix or flowchart, is that based on like known morphological relationships, like the example you gave of the mouth guarding and the size of the fish? Where does the information that goes into that vary?
Yeah, that was just kind of it should be expertise, and it was kind of me just kind of making it up. But and again, you can do model selection, and I made it up based on like having read theory, like Mengel again, like you could derive Beverton derived sort of like a M over K, LMAT over L infinity, and Mark also presents it in his book. And the derivation of those usually assumes that M is fixed exogenously, and then it optimizes the timing of mortality conditional on that. And so a lot of our theory has this sort of flowchart built into it. And so I'm not saying it's right, but I just tried to represent sort of what I think people have assumed implicitly before.
There's a question online from Reza Goljani, a postdoc at EEB. Is it possible to account for imperfect detection in SEMs?
Yeah, and so the next step is this. I'm merging this with my software VAST. So I've got a package called TinyVAST that I'm putting out on CRAN soon, and that's sort of like a multivariate spatial temporal model that simplifies notation interface using SEM notation. And so that's, for example, a place where you could put in, it basically has GAM MGCV notation. You could put in covariates, density and catchability covariates in GAM, and then it has an additive component for SEM, the separable space by SEM, and then a separable space by DSEM. And it just adds those together as a linear predictor and fits it as a big glim. So that's my current thing. Yeah.
Great. Any other questions?
Let's thank Jim.